Okay, first, I'd like to introduce Dr. Mark LeChevallier. He is the principal and manager of Dr. Water Consulting, a part-time consulting business he started after retiring from 30 years at American Water as Vice President Chief Environmental Officer. Dr. LeChevallier has authored over 300 research papers and received numerous awards for outstanding contributions to the science of water treatment. He is a fellow of the American Academy of Microbiology and the National Academy of Engineering. Sherry Williams has been employed at Newport News Waterworks as the Chief of Regulatory Compliance for the last 40 years. She has an MS in Chemistry from Old Dominion University and a BS in Environmental Science from Florida Institute of Technology. She is an AWWA honorary member and a Fuller Award recipient. She has served on various VA section committees, the VA section board as chair and secretary, and on the AWWA board of directors. She has participated in other WRF projects that investigated the fate of HAAs in the distribution system and the effectiveness of flushing to reduce lead. Next up is Sharon Sweeney. And Sharon is dedicated over 30 years to the water industry, working on rural water and sanitation issues as a Peace Corps volunteer, assisting water systems in Arkansas, and handling all phases of drinking water production and regulatory compliance. She has a deep understanding of source water, treatment, distribution, and water quality, along with regulatory and environmental concerns. Sharon has presented at various conferences and workshops and collaborated with organizations like the American Water Works Association, the CDC, the USGS, and the Water Research Foundation. Sharon holds Arkansas Class 4 water treatment and distribution operator licenses and has earned a Master's of Public Health, a Master's of Science in Interdisciplinary Science and Math, and she has a graduate certificate in conflict mediation. And last but certainly not least is Patsy Root, who is a Senior Manager of Government Affairs here at IDEXX Water. She has over 18 years of experience in ensuring laws, regulations, and guidance will protect public health. And she has also published numerous papers focused on addressing the risk of Legionella pneumophila. So our format today, Dr. LeChevallier will begin the webinar by providing an overview of the WRF 5156 study and its findings. During the second part of the webinar, Patsy will lead a conversation with Dr. LeChevallier, Sherry, and Sharon about the impacts of this study. We'll conclude the webinar with a Q&A session where all four panelists will answer your questions. So with introductions out of the way, I would now like to turn it over to you, Dr. LeChevallier.
Happy to talk about this topic. I've done a lot of different talks on Legionella, but this is the first time to the Klingon homeworld. So nice to know that we're going to have Legionella control on the Klingon homeworld. To understand the origin of that, you have to go to read through all of the where people are from in the chat list. Anyway, this first slide addresses the question, why are we talking about Legionella in the first place? You probably have seen some data since the year 2000. There's been a tenfold increase in the incidence of Legionella outbreaks in drinking water systems. But unless you kind of follow the CDC publications, about the middle of last year, they published an update from 2015 to 20,000 to the year 2020. And that data showed that 92% of the drinking water outbreaks, 60% of the cases, 97% of the hospitalizations, and 97% of the deaths due to waterborne outbreaks in that 5-year period was due to Legionnaires Disease. So that's pretty shocking that pretty much if there's an outbreak occurring in a water system, it's probably due to Legionella. So that information, along with the overall trend in the last 20 years, increases is the reason that we're having this discussion in the first place. Really risen to being kind of the key waterborne pathogen. And you ask, well, is Legionella regulated? Yes, it's regulated under the 1989 surface water treatment rule, but there are some problems with that. Most outbreaks are in buildings, not in distribution systems. And the surface water treatment rule doesn't apply to groundwater. And EPA published a couple of years ago this piece of data that said about a third of the outbreaks are in water systems served by groundwater, which don't necessarily have to meet the minimum disinfectant residuals in the distribution system like surface water systems. Perhaps you're aware that in Grand Rapids, Minnesota, not Michigan, that's the bigger name, unchlorinated groundwater system had a Legionnaires outbreak last year. And actually, we'll be talking about that outbreak and the great response that the utility did and how that outbreak was resolved at the WQTC this year. Actually, at WQTC. Also, the problem with the surface water treatment rule is that it requires maintenance of a disinfectant residual in the distribution system. It says that disinfectant, it requires a minimum of a detectable residual, which is not defined. In fact, a study done in Colorado found that when they gave operators dechlorinated tap water, they got results everywhere from 0 up to 0.9. So Colorado decided that anything less than 0.1 was essentially the same as detectable. So, there can be errors in that measurement. And of course, I think one of the big problems with this rule is that 5% of the locations in a distribution system can never have a residual and still the utility meet that regulation. Now, EPA is in the process of updating that surface water treatment rule. Supposedly, it's supposed to be out by July this year. I think with all of the things going on in the federal government, that might be delayed, but we'll see that, at some point in the near future, changes, and whether or not they're going to change this 5% part, I don't know. But, maintaining a disinfectant residual, an effective residual throughout the system has always been a principle of effective microbial control, whether it's Legionella, coliforms, or other microbes in water. So we're going to talk about this project 5156, and I'm only going to give you the thumbnail overview of that. The full report is in the chat. If you're interested, download that. There's a lot more details that I'm not going to cover. But the Reader's Digest version of that is that we enrolled 57 utilities to volunteer. These were brave utilities to volunteer and did the testing, most of them themselves, to collect a minimum of 3 samples per week for 12 weeks during the warm water months when water temperatures were greater than 15 degrees centigrade for 2 summers. So they are involved for 2 summers to do this testing. That project resulted in more than 9,000 samples. The utilities were scattered across the United States and included utilities with surface and groundwater supplies that maintained free and chloramine residuals in the distribution system, and had utilities of various sizes, geographical distribution. The utilities monitored for Legionella using LegAlert. We'll talk about that in just a second. But we also did some additional analysis by qPCR and confirmed the occurrence of Legionella in the LegAlert test by qPCR and MALDI-TOF, which is a technique for speciating Legionella in clinical laboratories. We also did some conventional culture and analysis by qPCR. I'll simply touch on that to show you why the LegAlert test was working so well. And we had a workshop on developing protocols of how to respond to Legionella in distribution systems. Mentioned that the utilities did this test, LegAlert. It is a 100, we use the 100 ml format. There is a 10 ml format in this quantity tray. So basically, the procedure is the quantity tray has 6 large wells and then 90 small wells and generates a most probable number, or MPN. some people get the idea that NPNs aren't very accurate because there used to be a 5-tube NPN test for coliform analysis, but with a 96-well NPN, it's actually very, very precise. So the overall test is pretty simple. You collect your water sample. You have to test and adjust the hardness is the only kind of ripple to this. But otherwise, you collect the water sample, you add the secret sauce, you pour it in the quantity tray, you put it in the sealer, almost like a seal meal, and then you put it in the incubator. So if you're doing the coliform test for total coliform and E. coli analysis, you have about 90% of the skills necessary to do the Legionella test. The only real big difference here is that the Legionella tray is incubated at 39 degrees. So to do the Legionella test in your lab, you'd have to have an incubator at 39 degrees, and you have to have the quantity tray sealer. So the only real technical differences between doing Legionella and coli tests. In our study, we found that 96% of the positive trays we could validate by detecting Legionella pneumophila within the trays. We had some utilities. There's a bit of a learning curve in using this test. On the far right, there's a positive control, and you can see the typical kind of brown color that Legionella pneumophila produces in this test. And the 3 trays right to the right of that are kind of light colored, and then there's kind of a dark, kind of almost a black colored tray. So these are some kind of atypical colors. Utilities were very anxious to find Legionella in the water system, so there's a bit of a learning curve. We end up finding that those 3 trays were not positive for Legionella pneumophila, the kind of atypical colors. Now the last tray on the far left has one tube of a kind of a brown color that, by all intents and purposes, looks like the positive control. Now, in our study, for maybe various reasons, we did not detect Legionella pneumophila in that tray. But even if the utility counted that as positive, it would only be an NPN of 2 per 100 mils, so a very low occurrence. Overall, for research, we wanted to detect Legionella pneumophila within the trays, but even if you took the results straightforward without any kind of subsequent testing, the results would be essentially very equivalent. So 96%. The Legionella test works very well. We did some comparisons. It was not the principal reason for this study, but we did some analysis between Legionella, the standard ISO method for Legionella pneumophila that many commercial laboratories would do, and a qPCR test that's now available from Hygienia that also measures viability qPCR. When we did the test, we had one sample positive for 143 samples. We had 1 positive by Legionella. We had none positive by the ISO method, and we only had 2 positive by the qPCR. So the 3 methods for measuring Legionella pneumophila gave very equivalent results, not a significant difference. The qPCR also measured Legionella species, and we found that 37% of the samples in drinking water distribution systems had Legionella species, and there was no correlation between detecting Legionella species and Legionella pneumophila, and that's shown in these other. And when we compare those results to other published results, our detection of Legionella was very similar. Legionella pneumophila was very similar to other studies, but using the viability qPCR, our results are about half about what other researchers have found for detection of Legionella species. So in the handouts, there's a paper I recently published arguing why I think for water utilities and public buildings, Legionella pneumophila is really the right target to be focusing on. And I'll let you look at that paper and see whether you kind of agree with that logic. So let's get to the results of these utilities. Of the 57 utilities that participated either by doing the analysis themselves, some provided historical data. We had 9,181 samples that were analyzed. Of those, almost 9,200 samples, we had 109 positives. So that results in about a 1.2% occurrence rate. But we had positives from 18 of the 57 utilities, so about 32%, almost a third of the utilities had some detection of Legionella at some point during the study. The occurrence of Legionella in these systems ranged from, well, obviously, 2/3 of the utilities had all zeros, but many times when Legionella was detected, frequently the numbers were 1 or 2. About 3/4 of our results were less than 10 per 100 milliliters, and only 5 results were greater than 100 per 100 mil, or 1 per mil. And show in a moment, we kind of use that as an action level to be taking extra action. So when Legionella occurs, it's very infrequent, about 1.2%. When it does occur, it's usually at very low levels, and only infrequently, over 9,000 samples, only a handful of times did we have levels at Legionella that we'd say really requires the utility to have some kind of, a response to that by flushing or increasing disinfection. And most of the times when the utility had a positive, they repeated that analysis at that same point of the next week after getting the positive Legionella tray, and almost always those repeat samples were negative. Only a handful of times that indicate there was some issue maybe with that sampling location. So it says that Legionella levels in distributed systems are low and transient and infrequently positive. These results show for those 18 utilities that had positives, most of the time that occurrence was less than 5%. So we kind of think that, that might be another tool that utilities use, that if I have occurrence of Legionella more than 5%, then that's kind of unusual. And often if utilities take action, they can bring that 5% rate down by maintaining a minimum disinfectant residual and by flushing the systems. Most of the time that when Legionella occurred, it occurred at temperatures greater than 16 degrees. So you can see the top panel is for the free chlorinated systems, and the bottom panel is for the chloraminated systems. We had some positives that are the dark colored circles in free chlorinated systems when temperatures were low, probably indicating that Legionella is simply surviving in the system. They're not really growing, and the only evidence of growth when levels are higher is when the water temperatures are warmer. This slide shows for free chlorine at the top and chloramine at the bottom, the relationship between Legionella concentrations and the concentration of disinfectant. And it shows when free chlorine was low, between 0 and 0.2, or chloramines were low, between 0 and 0.2. A higher percentage of samples were positive when the chlorine residuals were in that category than they were higher. So we'd recommend that utilities strive to maintain at least a 0.2 milligram per liter disinfectant residual, regardless if they are a free chlorinated system or a chloraminated system. There is a minor benefit from maintaining residuals between 0.2 and 0.4, but above that, you can see that we had detections of Legionella pneumophila even when free chlorine levels were greater than 1 milligram per liter, and in some cases when we had chloramine residuals were 2 or 3 milligrams per liter. So maintaining a residual doesn't guarantee that you won't have detection of Legionella. pneumophila in water systems, but you, if not having a residual, means you'll probably have it more frequently. So to maintain that low level of Legionella occurrence, maintain at least a minimum of 0.2 milligram per liter. That's a conclusion from this study. So the biggest benefit was maintaining 0.2. Next was 0.4, but boosting chlorine residuals to a lot higher levels doesn't necessarily has a diminishing return on that benefit. And you still can have Legionella even at high residuals. I mentioned that we had a workshop to talk about response protocols, and we had people from the research team, the project committee. We had people from the EPA and CDC. We had people from state health departments, international, because we had Canada. I won't get into the whole tariffs and trade thing with Canada, but anyway, we're great to have that participation, but including Paul van der Wielen from the Dutch and the Netherlands are doing a lot of research and regulations on Legionella. The Association of State Drinking Water Administrators, the state and territorial epidemiologists, water utilities, academics, and people from IDEXX and other people participated. The result of that was to develop a protocol to respond to Legionella in water systems. And I know this flow diagram looks complicated, but if you stop and kind of think about it, it's how we would respond to any bacteriological result in the water system. Anytime we got a positive, we would first ask, did we have the correct QA, QC in the sample collection, in the analysis, in the shipping, in transport of the sample? The next question is what we would probably do a repeat analysis. So we had a follow-up analysis in our Legionella. Based on that follow-up analysis, we would examine that data, and based on that result, we would take some action. So the action that we came up for the purpose of this research project, and again in the handouts there is this paper that describes this whole protocol, and we put that forward for utilities that are doing testing. Say, well, there's no guidance currently from EPA or CDC to health departments. Hopefully they'll be coming at some point in the future. But now you could at least follow, well, this is what we used in this research project. If the results are less than 1 MPN per mil or 100 per 100 milliliter, and the water chemistry and water quality and the chlorine residuals are normal for the system, and the repeats are negative, then that's kind of what we found from most of our cases. And you just continue to do the monitoring, no further communication. If it's greater than 1 per mil or 100 per 100 milliliter, then take proactive steps to flush, disinfect, and remediate any conditions you might find in that. We found that regulators, some are interested in the project and they wanted to have that result, and so we might inform them that we had a hit and these are the corrective actions we were taking. If the levels are greater than 10 per 100 milliliter or 1,000 or 1,000 per 100 milliliter, which never happened in this study, then we would consider that evidence that Legionella is not being controlled in the distribution system, and now would be a time to consult with your public health officials. That's never occurred, not in this study or in the prior studies that I've done with water utilities. So, not saying that it can't happen, but if you do monitoring and reasonably well operated system, that's not going to happen. So this is just guidance. This is it's not regulation. Legionella is not regulated at this point, but these are some guidelines to help utilities that want to do monitoring to kind of put these results in some kind of context. So let's last thing I'll talk about is putting this in a public health framework. And in 2019, there was this paper by Carrie Hamilton that did a risk analysis, a quantitative microbial risk analysis for Legionella. and did this analysis both based on what's called CSI, clinically significant illness. So these would basically be disease, thinking of cases of Legionnaires disease or Legionella infection, and did a calculation of how many Legionella would cause an infection, both on an annual basis and on a per exposure. So you often get one sample. You want to know, is this important or not? You know, how should I respond to that? Well, this paper was really helpful in putting that into that context, and I'll run through those numbers in just a moment. And did that analysis for both faucets, toilets, and showers, both conventional and for high efficiency, because if you have that low flow toilet that actually produces less aerosols, and showers, high efficiency showers may have different kinds of droplets. And so that CSI is the cases of Legionnaires disease, and then also did on an annual probability of infection. So this risk of infection now encompasses sensitive populations, maybe immunocompromised or elderly, that are at greater risk of incurring Legionnaires disease. And you can see how these arrows now are about 1,000 times, maybe a little bit less, but almost 3 orders of magnitude. So if we're worrying about whether it's causing disease or causing an infection, it has about 1,000-fold safety factor. And when we put those numbers into context, we end up with, I'm going to go back because the numbers were taken off of this. You can see that it is about, on a per sample basis, it's about 10 to the 5th, or about 1 per mil, or it's actually about 10 to the 6th. So it's that 10 per 100 mil is about a 1 in 10,000 probability of causing illness, and it's about 1,000 per mil. So about a 1 per mil has about a 1 in 10,000 probability of causing an infection. And EPA's used this 1 in 10,000 guidance in regulations for Giardia, Cryptosporidium, and viruses. So we're starting to put these numbers. You can see the guidance starts to put how EPA might be thinking about a regulation at some point in the future. Now, the assumption is that we were monitoring Legionella in distribution systems. All these samples were from distribution systems, not at sampling taps. They were flushed or collected from dedicated sampling. So we're assuming that the levels that are in the distribution system is flowing through the building and to the point of showers or toilet flushing or faucets. And people generally don't do that out on the street corner. So we're making that assumption that these levels are not changing as they go through the building. But when we put if we put all that data we just monitored from utilities into context, we found that even the utility that had a 15% occurrence of Legionella is still way below that 1 in 10,000 risk of infection for Legionnaires disease. So even at the levels we're finding in the water systems, these levels are below what EPA would typically regulate for water utilities. And for those utilities that had less than a 5% occurrence, which might be a kind of a guideline if you're trying to monitor and say, okay, I may occasionally have 1 or 2, but I don't want to have, Legionella above 5%. Those risks are almost at below the 10 to the minus 6, or almost 2 orders of magnitude lower than what EPA would be regulating. So at a very low risk from drinking water. So to kind of wrap this up, a national estimate would be exceedingly small, because remember, 2/3 of the utilities that did the monitoring never had any positives, and so their risk would be way below 10 to the minus 7 annual risk. And even the highest single concentration that we found would be less than a 10 to the minus 4 risk of infection for exposure, like from a faucet or a toilet. So I think these are good news to utilities that open the doors. The utilities now can go ahead and monitor Legionella and manage Legionella occurrences in their water systems, know that they are managing at below a public health risk threshold that would be used, and so you can find Legionella, you can manage it, you can control them well below these risk thresholds. So to wrap it up, utilities are finding it feasible to monitor. We had 57 utilities that did this for 2 years. The guidelines are critical, and we've published some guidelines that might be useful. We encourage EPA and CDC to pick this up, or public health departments, so that opens the door, that we can monitor and manage Legionella in water systems. The results of the study show that it's rare in properly operated systems. But to maintain an effective level of disinfectants is helpful, but not a panacea. Flushing, cleaning storage tanks, cleaning distribution lines are also important. Practice a good management, we just said, and the risk is low and properly managed utilities. Now, poorly managed utilities generally don't participate in research, so we can't say this occurs for every water system in the United States, but for the well-run systems that are trying to maintain a good job, this is probably representative of the kind of water that you have in your systems. So with that, there's my contact information, and it's also included in all of the material. If there are any questions, well, we're going to get to that in a second here. But with that, I'll turn it over to Patsy for our discussion.
Thank you, Mark. This is such great work. And in between this study, with Sherry and Sharon and all the other participants, it's really foundational, right, to our understanding of what can be done. And excuse me, that it shouldn't be looked at as a scary thing. So I'm really happy to have Sherry, Sharon, and Mark, and we're going to go through a panel discussion. We have first question. So Mark, I think I'm going to direct this to you. I'm just going to keep you talking through the whole thing. Sorry. If the exposure risk really comes from the buildings, what do you think is the strongest argument to the senior utility folks that would convince them to proactively manage and monitor Legionella pneumophila in the distribution system?
Exactly. I just said, the risk is low. So you could say, oh, okay, I don't have to worry about this. I'll focus on PFAS or lead or whatever. I don't think that's the takeaway I would put forward. First of all, that was for the 57 utilities that participated in the research project. That may not be your utility. And even in the utilities that did participate, we found that there might be zones of your system, particular locations that that could be problematic. So, so just saying that I don't have to worry about this, because still 92% of the outbreaks are due to Legionnaires disease. But and so people have been afraid to do the monitoring. That might be another reason. But now we can show you we don't have to be afraid. The other utilities are doing that. In fact, it's really being beneficial to be on top of this issue, because if an outbreak occurs, and it's probably, like you say, going to occur in a building, invariably that's going to ask questions. Well, Legionella is in water or in our building. That building comes from your system. What do you guys know about it? Well, we don't know anything. We never tested. Maybe some other researchers do that, but our utilities never looked at this. That's not a great answer to give. The better answer is, we looked at this. We've done some monitoring, and we know that Legionella levels are well controlled below risk levels. And so, to be knowledgeable on top of that is a better answer. And to say, well, we're not required, we're not regulated, we don't know, is not a great answer. People are worried about having that may be too risky. Well, not testing not knowing is not going to put you, alleviate you of liability. So
Exactly.
knowing the top of the issue is really beneficial.
Thank you, Mark. Let's go to the next question. Let's see. Challenges and lessons learned. What were the biggest challenges or unexpected outcomes your utility faced during the study, and how did you overcome them? Hey, Sherry, do you want to tackle that one first?
Sure. Just initially is trying to determine where we're going to sample from. We're a peninsula, and we do have a lot of dead ends, and we do have ongoing flushing. So when we were selecting our locations, we were trying to get in areas that we were not flushing in. And so that's what we kind of focused on, low chlorine areas that we were not flushing. The biggest thing in the analysis challenge is the sampling schedule, because with this test having such a long incubation, and the rule is not to peak during those 7 days, we had to be sure that we collected all the samples we wanted to collect on the one day. That was a challenge. The rest of the monitoring that we did along with this study were things that we were already doing. Our practices, any location that has a residual of less than 2, we do the full nitrogen series, HPCs, pH, etc.
2 or 0.2?
2.
2. Okay. Just wanted to be sure.
Because they're a chloraminated system.
Yeah.
Yes,
That's
chloraminated system. So yes.
Yeah. So let me see. I got to go back to 62 here. Previous. Bear with me, folks. Sharon, what did you think about the results from the study? Did they help shape your approach to Legionella monitoring?
Well, I was, of course, pleasantly surprised. I suppose initially I thought there might be a higher detection rate, so it's always good to know that even in areas of the system that might not be as optimally turned over as we would like, to not have detections was great. As far as, I guess, how that helped us operate our system, we could perhaps relax a little bit with our chlorination practices. We were trying to really boost chlorination, and learning that 0.4, 0.5 for free chlorine is adequate. That was quite helpful.
Yeah. Mark, I remember listening to you do a presentation one time. Excuse me, and hang on. And you saying that disinfection isn't necessarily a silver bullet. And I think that was really sort of revolutionary for other people in the audience. So,
well, and
sorry.
To kind of, Sharon's a little modest, because they actually put a lot, and you published this in the OpFlow, as I thought was telling somebody earlier today, was great work. After finding that there was parts of the system that were low, you really focused on turnover tanks and kind of redirected your capital program to that, so that you were more consistent. And not only made a more consistent residual, which then after that you never had Legionella problems in your system, but you also lowered your THM levels, which most people think, if I have to boost chlorine residual, what am I going to do with the THMs? But you actually had a great result by kind of removing the dead spots and having less detention time in your system. It actually had a benefit from the THM residual as well.
That's awesome.
Maybe you want to talk
Yeah. Okay.
Yeah, that was a great outcome. Not focusing on one single item.
Yeah. That's why it's a system. You have a lot to pay attention to in the system, and dead legs are definitely one of them. But managing your disinfectant level is also pretty important. So the next question I have here is about future planning. Are there any long-term strategy changes at your utility that you've made or will make based on the study's findings. Sherry, you want to do that one first?
No, not really. I mean, we have a pretty good program just because of our chloramination and watching nitrification. We did not continue doing Legionella testing last year, but we are considering possibly bringing it back in a limited amount this summer.
Mm-hmm. Cool.
I would say the one thing I shared with the group is the one location that we seem to constantly get a positive hit, we did determine what the problem was, and our distribution folks actually rectified it. The pipe was apparently not buried deep enough, and so that particular location, the temperatures were quite warm. And once that was taken care of, the result seems to improve.
Isn't that interesting? Very cool. Impact on operations and processes. How have the findings helped you shape capital programs in your organization? Sherry, do you want to start and then have Sharon jump in?
Again, participating in the study really hasn't had any impact on our operations and process. We're not increasing our chlorine. What's leaving our treatment plants is kind of high enough, and we're not at this point planning any major changes to tank operations. We are continuing our seasonal flushing for those locations that we know that are low residuals. So not really much of an impact, but we're very happy to participate in the study.
Yeah, I'm sure it was probably very revealing. Sharon, you have anything you want to add to that?
Well, as Mark referenced earlier, we focused more on holistically managing our system. We invested in tank mixing, and we found that to be beneficial. It was very cost effective and a great way to maintain the residual throughout the tanks and the entire distribution system for a relatively small amount of capital funding.
Cool. Good. I think we got time for... Oh, go ahead. Yeah, please.
And, the fact that we got on the chlorinated systems residuals below 0.2, measurements below 0.2 meant that the utilities were going out and targeting Legionella monitoring during nitrification. So nitrification control, as Sherry indicated, has the double benefit of having, Legionella management as well. Because otherwise, in our first study, we didn't have any measurements below 0.5 milligrams per liter in chlorinated systems, and no detections at that low level. So it said that utilities in this project did a great job in finding those kind of problem locations. And but anyway, the nitrification control doubles as Legionella control as well.
Awesome. Good point to make. Thank you so much. It looks like we have a lot of questions queuing up, so I'm going to move to a couple of very short polls, and I'm going to solicit Clint to help me with those. If you all would participate, it would be greatly appreciated. We'd love to hear where your thoughts are right now. The first poll is, how concerned are you about the risks created by potential Legionella pneumophila in your drinking water system? We'll see where everybody lands here. How concerned are you about the risks created by potential Legionella pneumophila in your drinking water distribution system? Looks like most people are somewhat concerned. Hopefully the information that you've heard from Mark and the study participants will give you some calming and better feeling about that. Let's go to the next question, the next poll question, Clint. Second question and last one. Do you believe your utility managers and decision makers understand and agree on the best way forward? We'll wait for your responses and we'll take a look at those. Looks like It's pretty split here. Yep. Thank you for voting. Appreciate it. It's nice to see where people stand. So it looks like a few are still unsure about alignment and understanding with their utility managers and decision makers, but there's still a good amount of people that feel clear about it, or no differences. Awesome. Thank you very much. Appreciate you guys participating in the polls. So now we are going to move to the question and answer period. Excuse me. And it looks like we have a lot. So Will is asking, where were the samples collected? From interior plumbing or somewhere else, such as a dedicated sample tap outside of a building? Mark, do you want to start with that?
Yeah, I mean, you can. There's lots of data in the full report, but the short answer is that all the samples were flushed. So we wanted, like we do for total coliform, so we wanted water that's represented in the distribution systems. But over half, I think 56% of the samples were from dedicated sampling stations, and there actually wasn't any difference in occurrence between the sampling stations and the ones that come from taps. A few utilities collect samples from hydrants. I wouldn't recommend, but that's their routine procedure for doing it. So we use the routine. Hydrants were slightly higher, but it didn't really influence the overall. But this study was representing water in the distribution.
Gotcha. Thank you. Another question comes from Manly. They ask, can you please speak what approaches are available to remediate Legionella contamination inside the building if the regular flushing and disinfection doesn't work? I'm going to start by saying, if you had a water management plan based on the ASHRAE 188 standard, you might not need to remediate, but which of the three of you would like to address that? Speak to the approaches available to remediate Legionella contamination inside the building.
I'll just say that this study really focused on the utility portion. I mean, there's lots of data, a lot of papers and guidance and stuff on building management, like you just mentioned, and so if that's happening in a building and not in your distribution system, then it's really up to the water management plan to look at that. Flushing, maintaining residual, looking for dead legs. There's lots of guidance. CDC is a place to go to. ASHRAE is a place to go to. You can find a lot of that on the internet.
Yeah. Yep. Another question from Jarrett. He's asking, did any utilities conduct sampling at times and locations of specific maintenance activity, such as stoppage of water flow for repair or other activity? If yes, were those test results different from periods of normal activity? Sherry or Sharon, do you have a response to that?
We did not. We did it under normal operations.
Yeah. And Sharon?
We also did. We didn't specifically target periods of outages or whatnot. We combined the sampling with our routine coliform sampling.
Yep.
With that, sometimes we did sample in an area that had a main break or work. It wasn't planned, but sometimes we did happen to catch those events, and we didn't see any difference in results.
Okay. Thank you for that. Let me see. John is asking, as Mark pointed out, in buildings like hotels, water can stagnate in pipes for extended periods. This delayed usage, sometimes months later, likely explains the yearly Legionella outbreaks. How can you educate on this?
And that is a great question. And I think that was the other reason that utilities should be on top of Legionella and do the monitoring, is that they can set example for their customers. So they could say, well, we're knowledgeable about that. So there's the Water Research Foundation has a project on communicating about opportunistic pathogens in building plumbing. So there's a lot of information in there on setting up web pages and communicating with customers. Before I left American Water, they had a program particularly reaching out to their industrial and commercial customers. So those are customers that use large volumes of water. They're your best customers, so you want to take extra care of them. And so that was a great, for the customer services that are reaching out to them to say, well, what do you know about Legionella? Do you have a water management plan? This is that you can go to our website and you can find information. So I think, being on top of this as a utility puts you in a position to better communicate, particularly to your industrial commercial customers.
Agree. I agree. Ross has a question. He said the slide went by quickly. Does the data show if chlorine or chloramine is a more effective disinfectant? Mark, do you want to grab that one first?
There's a lot of research around the two. So for starters, both of them, and ozone and chlorine dioxide, all of them will be effective, and they all kill Legionella. So they're all, you manage your system well, that will work. There's probably a dozen papers that show for hospitals that are fed chloraminated water, they seem to have lower incidence of Legionnaires disease than hospitals with free chlorinated water. There's some interesting reasons why that. We actually have hypothesized that in a 2018 paper. So there's still some reasons. But if you have low chloramine or low free chlorine, as we just showed you, Legionella can occur. So I think the literature says there's probably a slight edge to chloramines for Legionella control. Free chlorine has some other benefits for other microbes. So it's a little bit balancing. But the answer is, if you look at all the data, chloramines probably have the nod for Legionella.
Okay. We get time for one more question. Adam is asking, were the utilities that showed positive samples asked to report their flushing plans from before and after the test was taken? Sharon, do you have a response to that? Or Sherry?
Sure.
So the utility, about flushing, before and after the test was taken.
We didn't send any plan to the researchers, and in this particular study, we did not have a positive result in a previous experience we had, and we did supply our flushing and our remediation plans to the research team when that occurred, as well as to our regulatory agency.
So the first part of what you said was pretty interesting, because you participated in one of Mark's previous studies, and then you participated in this larger study. So you said, say what you said in the beginning again. In the first study,
I'm not sure.
You had some positivity, but in the second one you didn't, right?
Correct.
What happened between that first study and the second one?
Well, we applied the information we learned, and we improved our distribution system management practices.
And that's what research is all about.
Yeah, absolutely.
How can we do things better, right? So I very much appreciate everyone being on the webinar today.